When a cell undergoes division, it starts an interphase. Interphase is 90% of the cell cycle. In interphase, the cell carries out its function, but also grows, replicates DNA, and prepares for division. The first stage of interphase is G1. G can be thought of as growth. In the G1 phase, the cell undergoes vigorous cell growth. It doubles organelles, acquires necessary materials for DNA synthesis, and gains in size. G1 is a checkpoint where the cell checks for cell size, nutrients, growth factors, and DNA damage. The next stage of interphase is called the S stage. The S stands for synthesis. In the S stage, the cell grows and DNA is replicated, therefore the chromatin doubles. The third and final stage in interphase is the G2 stage. In G2, the cell grows and does final preparations for division. G2 is a checkpoint where the cell checks for cell size and DNA replication. At this point in the cell cycle, the cell has completed interphase and is now entering mitosis. Mitosis is the division of the nucleus and genetic material. The third and final checkpoint is in mitosis, and it checks for chromosome attachment to the spindle. If chromosomes are not properly aligned, mitosis will not continue. The first stage of mitosis is prophase. In prophase, the nuclear membrane disappears, the nucleolus disappears, chromatin coils into chromosomes, centrosomes start moving towards opposite poles, and mitotic spindle fibers start appearing between the chromosomes. The next stage in mitosis is metaphase. In metaphase, kinetochore fibers, which are attached to the centromeres, pull chromosomes toward the center of the cell. The next stage in mitosis is anaphase. In anaphase, chromatids separate at the centromere and move toward the spindle poles. This way, each pole receives the same number and kinds of chromosomes for when the cell divides. The next stage of mitosis is telophase. In telophase, two daughter cells that are exactly identical are forming, and the nuclear envelopes and nucleoli reappear. Chromosomes reach opposite ends and return to chromatin. The fibers disassemble and spindles disappear. The final stage of mitosis is cytokinesis. In cytokinesis, the cell splits into two identical daughter cells, each having the same number of chromosomes as the original parent cell.